Hello, welcome to 13 Universal Learning Beyond Barrier. This is Shedro, your biology teacher, and we're going to be looking at A-level CIE biology. Okay, all topics series 5. We're still on the MCQ series. Very soon we're going to go into your paper 2. We'll bring into you paper 4, paper 3, 5. Okay, but we're dealing with MCQ. So let's dive in. Get your pen and your writing pad. Let's start. Which statement about light microscope is correct? Which statement is correct? Okay, we're looking for the correct one. As the resolution increases, the magnification increases. Is this correct? As the magnification increases, the image always becomes clearer. Is that correct? The resolution will decrease as colored light of increasing wavelength is used. All right, which one is correct, guys? Okay, let's pause this video and try to solve it. Pause it and try to solve it. Okay, but let's analyze this, shall we? As the resolution increases, the magnification increases. This is not true. Resolution deals with details. Okay, resolution deals with details, while magnification deals with size. They are not correlated positively. As a matter of fact, as magnification increases to a particular point, resolution, right, starts decreasing. So they don't have positive correlation. Resolution and magnification, they do not increase at the same time. All right. So when you increase resolution, it has nothing to do with magnification. Is that okay? All right. A is wrong. B says as the magnification increases, the image always becomes clearer. Like I said, magnification deals with size, not details. Now, if you continue to increase magnification, a time comes the image will become blurry. So the image is not always, clearer. At a particular point, at the boundary of resolution, of course, when you get there, as you continue to increase magnification, the image becomes blurry. So option B is not correct. Look at C. So the resolution will decrease as colored light of increasing wavelength used. Now, this is very important. Let's look at it. Remember that wavelength, is that okay? Wavelength is inversely proportional. Wavelength of radiation is inversely proportional to resolution. Yes, don't forget this. So the shorter the wavelength, the higher the resolution. The longer the wavelength, the lower the resolution. And that is why light microscope, the resolution is 200 nanometers. Okay, of course, because the wavelength, look at the wavelength there. All right, so it has low resolution because it has long wavelength. But electron microscope is 0.0, okay, 5 nanometers. Is that okay? Now, because the wavelength is small, the resolution is high. So this can see way more details because it can see details from 0.5 and above. This can only see details. This is light microscope. Okay, light microscope can only see details from 200 nanometers and above, while electron microscope can see details from 0.5 nanometers and above. So which one sees more? Of course, is this. Okay, of course, is electron microscope because it sees more details, right? From 0.5 and above. This sees from 200 and above. So this can see details from 199 down to like 1. This can see them. All right, so this C is correct. All right, so the resolution will decrease as wavelength increases. All right, that is this is the relationship. So of course, this is magnification and resolution are terms that relate to the same factor. They don't relate to the same factor. All right, they're not related. So the answer is C. Let's go to the next question. The diagram shows a stage micrometer scale viewed through an eyepiece containing a graticle. The small divisions of the stage micrometer scales are 0.1 millimeters. You can see that the stage micrometer scale is replaced by a slide of a plant cell. You can see that. All right, so what is the maximum actual length of the nucleus in the plant cell? Okay, so pause this video and try to solve it. So look at it, guys. Remember, what we're looking for is the maximum actual length of the nucleus in the plant cell. Where is the nucleus here? Okay, the nucleus is at the edge. That is it. Okay, this all these ones here, can you see this? All of these, they are the chloroplast. This is the vacuole, this white space here. The vacuole pushes both the nucleus and the chloroplast to the edge, so that chloroplast can absorb more light, but the nucleus is at the periphery. So look at it. If you check this, measure from here. All right, let's look at this. Okay, how many radicals from here? Let's look at it critically. So approximately how many from here? All right, if you go this way down, right, and then that way down, it's okay. So that's approximately 10 divisions. Is that okay? Approximately 10 divisions from here to here. Okay, count it. 1 2, so 10 divisions. All right, now but let's look at the calibration. You look at here, let's get calibration first. They told us that 1 division of stage micrometer. Stage micrometer, this is from here to here is 1 division. Okay, from here to here, from here to here, this large bigger divisions are stage micrometer. These ones are the eyepiece graticle. So from here to here is 0.1 millimeters. Okay, while from here to here is 40. 90 minus 50, that's 40. From here to here, 10, 20, 30, 40. Okay, so that means that 40. Okay, let's do it. That means that 40 eyepiece graticle, is that okay, equals what? Eyepiece graticle gives us 0.1 millimeters, okay, which is the same thing as 1 division. 0.1 millimeters. Now if you convert 0.1 millimeters, which is very important, convert it to micrometers, multiply by 1000, you will have what? 10 micrometers. So that's 10 micrometers. So guys, this 10 micrometers. All right, so that means that 40 gives me 100, no, not 10, 100 micrometers. Okay, because if you multiply 0.1 by 1000, remember, if you are moving from millimeters to micrometers, multiply by the factor 1000, that is 10 to the power 3. Okay, right, multiply by 1000. Okay, you have 10. Don't forget that. So that means 40 gives me 10. Remember, here we got 10 from here to here, 10 divisions. If 40 divisions gives me 100 micrometers, 10 divisions will give me what? 10 eyepiece graticle. What will it give me? Help me solve it. So that will be equal to 10 times, so it will be 10 times 100. All right, 10 times 100 all over 40. Very important. 10 times 100 all over 40. Okay, so this cancels this. Okay, and of course 4 divides 100 will give you 25. So that means the size of that nucleus, the real size, the conventional size is 25 micrometers. Is that okay? But the eyepiece graticle size is 10 divisions. All right, so these 10 divisions will give us 25 micrometers. So 25 micrometers is the size of this nucleus. All right, we just said 40 gives us, because from here to here is 1 division, which is 0.1 millimeters, and that is for stage micrometer. For eyepiece, from here to here is 40. So 40 gives us 0.1. Convert 0.1 millimeters to micrometers, multiply by 10 to the power 3, 100 micrometers. So if 40 gives us 100 micrometers, 10, which is that of the nucleus, will give us what? 10 times that 100 over 40. That gives us 25 micrometers. Want to hear your comment. So the answer is actually 25B. All right, so let's go to the next question. What is the correct order of the involvement of the cell structures in the synthesis and secretions of an enzyme? All right, an enzyme: cell surface membrane, Golgi body, nucleus, rough endoplasmic reticulum. All right, look at the options. So what is the order, guys? Enzyme is a protein. Remember, guys. Pause the video and try to solve. Enzyme is a protein. Okay, so during protein synthesis, P is protein. It's synthesized by, first of all, transcription, right? So I call it TC. Transcription will occur first, and then translation will occur second, of course, and then after translation, remember, modification has to come. The protein has to be modified. I'll call it MD modification. All right. Now, if it's going to be secreted, before we now talk about exocytosis. So if it's going outside the cell, exocytosis will be there. All right. So EX is exocytosis. So protein synthesis. The first process is transcription. So where does transcription occur here? Where? 3. Nucleus. So I go to the option. Where does 3 start from? C. That's my answer. Now after transcription, mRNA goes to where? Rough endoplasmic reticulum. 4, because it contains ribosome. For what to happen? For translation. Translation occurs on rough endoplasmic reticulum, the ribosome attached to it. So after it has been modified, after translation has occurred, where does it go next? To Golgi for modification. Golgi folds the protein, forms the tertiary structure of it, and then it goes to cell surface membrane, where it is moved out of the cell. So the answer is C. All right. Central cilia, microtubules, and microvilli are structures found in eukaryotic cells. Which statement about these cellular structures is correct? All right, look at the first one. Central cilia and microvilli are composed of microtubules. Is that correct? Central play a role in mitosis and semi-conservative DNA replication. Is that correct? Cilia and microvilli both increase the cell surface area for absorption. Microtubules are made of protein and form the spindle in mitosis. Okay, so it's spindle in mitosis, not spindle in. Okay, so which one is correct? Let's analyze it. Pause the video and check it out. Central cilia, central contains microtubules. Yes, made up of 9 triplets of microtubules in central. Cilia, of course, is made up of 9 duplets of microtubules with 2 singlets at the middle. Okay, microvilli doesn't have microtubules, so A cannot be the answer. Okay, B says central play a role in mitosis and semi-conservative replication. No, it doesn't play a role in semi-conservative replication of DNA. It does not play a role here, so B is wrong. C says cilia and microvilli both increase the surface area for absorption. Nope. What increases surface area of absorption is microvilli. Cilia is for movement. Microvilli increases surface area of absorption. So C is wrong. That means D is right. The answer. Let's check it out. Microtubules are made of protein. Yep. Tubulin protein. And form spindle in mitosis. Yes, they form spindle in mitosis. Yeah, the assembly of microtubules is called spindle. All right, let's go to the next question. So the answer is D. Consider question 5. Which description of plasmodesmata is correct? They are channels. Okay, so which one is the answer, guys? Let's walk through it. A says they are channels through plant cell walls lined by the cell surface membrane. Okay, is this correct? Okay, let's quickly draw this. Come with me, let's draw it. Let's draw this is plant cell wall. Okay, come with me. That's plant cell wall here. Yeah. Okay, now see, this is a cell linked to another cell, plant cell. Let me just see. Okay, can draw this with me. Pardon my diagram. My drawing tool is not stable, but just trying to create, all right, a picture of what we're dealing with here. Okay, now of course there's always vacuole here, I'm not interested in that, but watch here guys. This is plasmodious matter. Here is the link between this cell and this cell. This gap here is plasmodious matter. So A says there are channels through plant cell walls. Yes, so they move through cell walls. Okay, but cell walls are no longer there. Okay, and they are lined by cell surface membrane. So cell surface membrane is here, cell surface membrane here. This is cell wall, cell wall, cell wall, cell wall. So plasmodious matter here is here. It is lined on both sides, lined by cell membrane. Okay, lined by cell membrane. This cell membrane lining it. Cell membrane. This inner line here. That's inner line here. So A seems to be correct. Okay, B says there are channels through plant cell walls that are formed from protein. No, they're not formed from proteins. C says there are channels required for the movement of water through the apoplast. Water moves in apoplast through the cell wall. Cell wall is apoplast. Okay, plasmodious matter is symplast. All right. D says they can become lignified and form pits in the walls of xylem vessel element. That's not correct. Okay, so what is then the answer? The answer is A. Let us know what you are thinking, guys. Which cell organelles does not have nucleic acid? Chloroplast. Okay, look at it. Pause the video, tell us the answer. Which one does not have nucleic acid? Chloroplast has circular DNA. Okay, 70S ribosome, which has ribosome RNA. Golgi doesn't have it. Mitochondria has circular DNA. Ribosome has ribosomal RNA, which is nucleic acid. So the answer, of course, is B. B. Which processes occurs in eukaryotes and prokaryotes? All right, hydrolysis, mitosis, transcription, translation. Which one is the answer, guys? So mitosis does not occur. That's the only thing that doesn't occur in prokaryotes. Okay, because mitosis involves separation of course, chromosomes. Chromosomes are not in prokaryotes. Chromosomes include DNA and histone protein. The genetic material of prokaryotes actually is only DNA. They don't have histone protein. All right, for most of them. So mitosis does not occur there. It's only hydrolysis, transcription, translation. They all occur. They have DNA, so they transcribe their gene. They have ribosomes, so translation occurs there. Right? They have water and enzymes. Hydrolysis can occur. So 1, 3, 4. Which one is the answer? Is C. All right. Which bonds are present in all viruses? Okay. They keep repeating this question, guys. Covalent ester, phosphodiester. Which one is the answer? Tell me. Tell me. If you get it, I'll give you chocolate. All right, we have covalent. Now, covalent can be found. You know that viruses have protein. They have protein coat, capsid, okay, DNA inside or RNA. So proteins contain peptide bond. Peptide bonds are covalent, right? The covalent bond. Not all viruses have lipids, okay, which they take up from their host cell. Ester bond is formed in lipids. This can be found, of course, this is found in protein. This is found in nucleic acid, RNA, DNA. This is lipid. So all viruses must have, okay, they must have protein. Do you understand? Protein code, and they must have DNA or RNA. They must not have lipids. So ester represents lipids. This protein, this nucleic acid, DNA, or RNA. So it's 1 and 3. What do you think, guys? What do you think? So you see, the knowledge is very important. You have to memorize, understand, and then remember biological concepts so that you can apply them. Biology, all right, it's quite bulky, so you have to remember concepts you are taught. Look at this. The diagram shows the structure of a monomer. This is monomer, okay? Which molecule contains this monomer? Is monomer, not monomer. Okay, monomer. All right, it's a typo error. So which one? Oh, I mistakenly clicked the answer. But let's see. Watch. What is this? This is alpha glucose. Why? The OH attached to the first carbon atom, all right, is below the plane. This is the plane of the ring structure. This is the plane. If you divide this into two, divide it into two, okay, The OH attached to the first carbon is below. That's alpha glucose. So alpha glucose, amylopectin is a polysaccharide of alpha glucose. Glycogen is a polysaccharide of alpha glucose. Sucrose contains alpha glucose and fructose. So it's everywhere. That's why it's okay. That's why it's B. But this is alpha glucose, all right? First carbon atom, right? OH below the plane of the ring structure. Monomer of this, and part of sucrose. Sucrose is a disaccharide. All right. Okay, let's go to the last question. A triglyceride consists of glycerol and 3 fatty acids, linoleic acid L, oleic acid O, and palmitic acid P. The diagram shows one possible arrangement of the fatty acid L, O, and P in the molecule. So this is LOP. Okay, LOP. So what is the total number of different arrangements of the fatty acids in this triglyceride? So what's the arrangement? Okay, look at LOP. This is one arrangement. Okay, so what do you think is the answer? LOP, but you can also have, okay, you can have LOP, you can also have O, L, okay, P. All right, P can get then go in the middle. You can have the same O, P, L. So if you alter this, you get the same thing. If for example, if you put P, O, L, is the same thing as this. It's the same thing. It's the same thing as this pole. Either piece is down up. So this is the same thing as this. So if you alter it, so it's only 3 times. This 2. This is 1, 2, 3. A. So the answer is A. So thank you guys for hanging on. Okay, please kindly leave your questions, make comments, share the videos, and like the video. So bring you all topics series 6. Okay, and get ready for paper 2 very soon. Is going to be online. Thank you. Bye.